The next speaker is Kristina Wright. She is a behavioral neuroscience PhD student in the psychology department. She earned her bachelor's degree in psychology at Boston College while volunteering in Dr. Gorica Petrovic's neurobiology of feeding behavior laboratory. Before joining the PhD program at BC, she devoted 2 years to opening Dr. Michael McDonough's lab, serving as a lab manager and research assistant. During this period, she examined the effects of early life stress and adolescent alcohol drinking, adult fear using rodent models. As a graduate student, Kristina is using a combination of complex behavior testing, in vivo electrophysiology, and optogenetics to examine specific novel roles for brain regions in the appropriate acquisition and response to fearful stimuli. In the little spare time she has, Kristina enjoys classical ballet, drawing, witty cross-stitching, and boatloads of coffee. Let's welcome Kristina with her talk, The Power of the Brain, Finding Strengths in Neuroscience.
Sorry, last time they introduced me, I wasn't quite ready. All right, so today I'm going to be talking to you a little bit about how I got interested in neuroscience, why I'm passionate about it, and then what exactly I do in this lab, because even just listening to that introduction, it's a lot of big words that sometimes I don't even really understand. This is the first person I need to introduce you to in order to tell my story, and this is my mother. When I was about 8 years old, my mom had a brain aneurysm, and that's just a rupture of a blood vessel in her brain. And she went from someone who was loving and caring and a really wonderful mother to someone who couldn't hold a job, never really got her life back on track, and ultimately ended up getting really heavily involved in drugs, which ended her life. Sorry, this part is a little bit tricky for me. So as a kid, when you're about 8 or 9 years old and this is happening, you're not really sure how to interpret it, right? You can imagine you don't exactly understand the power of the brain, right? You don't understand what could have contributed to this. You're just kind of questioning why and what's going on. And so the very first thing I learned from this experience was just how powerful the brain was. And that was a bit that I could actually comprehend. The next step in that, as I got a little bit older and I realized that I was still really curious, was just understanding the power of the brain and being able to look at different areas of the brain and see exactly what type of behavioral responses they produce and how even a tiny little part of your body in this one organ has the ability to change your life forever. Disclaimer, which was already mentioned, I use rodent models. And a lot of people, the first question I get is, okay, Kristina, that's all fine and dandy, you play with rats, but what do rats have anything to do with humans? And I'm here to tell you that, generally speaking, we're looking at, in our lab, fear circuitry, or how people respond to fearful stimuli. And those are the types of circuitries that would be common across species, and we call them evolutionarily conserved. So what that means is, if you ask, or if you show an animal, a rat specifically, something that's threatening, they're going to respond to it in a similar way that a human would. And they're going to be activating similar areas of their brains. And using rodent models, we'll be able to go back into those areas of the brains that we identify and then potentially identify them as targets for treatment for disorders that are disruptions of that circuitry. So what I use to do this, to bridge the brain and behavior and to understand how those two things are related, are these devices called electrodes. And essentially, I take an electrode, which I'm holding up here, that I build in the lab, which I'm sure you can't see, but I promise you I'm holding something. And I implant it into a part of the brain that I think is really important to this fear circuitry, or these fear responses. And so then what I can do is, once this electrode is in the brain, I can plug it into a computer screen, effectively plugging a rat into a computer and say, okay, so I see that you're responding to this fearful stimuli, but what are those neurons in your head doing? What is actually going on at a neural level that's allowing you to produce these accurate and rapid fear responses to those stimuli? So some people want to know what this actually looks like. And here's an example of a rat that's had this surgery, where I implanted this electrode into a part of the brain that I think is important, and gave them a fancy head cap, a little hat to cover it so that it didn't get damaged. All right, so in order to bridge the gap between the brain and behavior, we need to be activating the circuitry that we care about in order to see what the neurons are doing. So what that means is we need to expose our rats to a situation that could potentially be threatening. And we do that by exposing our rats to 3 separate sound cues, one of which is always followed by shock, which is our danger cue, one of which is never followed by shock, which is our safety cue, and one of which is followed by shock 38% of the time. And that's our uncertainty cue. And so you can imagine if rats are discriminating between these cues the way that humans do, that they would assign a high level of fear to the danger cue, a low level of fear to the safety cue, and a moderate level of fear to that uncertainty cue over times of being presented with these stimuli. And that's exactly what we see here. With level of fear on our y-axis, we've got high fear to that danger cue, low fear to that safety cue, and the uncertainty cue fear falls somewhere in between. So this is that behavior part of the equation. We're activating the circuitry that we care about, and now we have an electrode, a fancy little device that's going to allow us to see what the neurons are doing while these animals are doing this task. So what do the neurons look like? Well, kind of like what you would expect. If neurons were to care about this stuff, then they're going to be caring about it at the time that it's happening, so that we know that the neural activity is related to the behavior that's going on. And that's what we see here. So if we look at the x-axis here, we've got the cue exposure, so just the animals listening to this cue. You can see there's a blip of activity right at the beginning during the first second. So that's the first second when these sound cues come on. And even more interesting is you can see that kind of graded amount of excitation that we saw in terms of the fear on the other side. So let me explain that a little bit further. What we're looking at on the y-axis here is firing, actual neurons firing. And when you play these sound cues, this is an example of one neuron that will excite a lot to the danger cue, excite a little bit less to that uncertainty cue, and excite even less to that safety cue. So this is the example of how the neural activity can map onto and help produce the behavioral responses that you would expect in these type of threatening situations. So, getting away from the graphs, at this point we've talked about how the brain has the ability to change your behavior. And we've talked about fear circuitry in the brain and how it produces this behavior. You can also imagine how disruptions in this circuitry can give rise to disorders such as post-traumatic stress disorder. Because if these are the neurons that are telling you how to react appropriately in this situation, and those neurons are not doing what they're supposed to be doing, they're not showing that graded potential, then maybe you're not doing what you're supposed to be doing. Maybe you're overexciting and being overexcited by something that's safe in an instance that would be consistent with post-traumatic stress disorder. Or maybe you're reacting inconsistently with something that's dangerous and not being bothered by it at all. And both of those things can be maladaptive. So the last thing in our lab that we do is not only looking at normal circuitry and then how this circuitry can be disrupted, but we're also looking at how environmental factors can change this brain circuitry. So we have models that we use in the rats that'll be looking at how adolescent stress and adolescent alcohol abuse can make changes in these neurons and make it so that you're not able to respond to these stimuli appropriately. And with that, the very last thing I want to do is introduce you to my grandmother, who is single-handedly the best single parent I could have ever had, who really helped me get through my mother's passing and helped me get to my place in neuroscience today. And finally, additional thanks to my PI, Mike McDaniel, who lets me play with a lot of really expensive equipment. Dr. Grisa Petrovich, who allowed me to step foot in my lab for the first time. And all of the other people who have supported me along the way. Thank you.